All right, so next up we need to talk about antimicrobial resistance, which is an absolutely gigantic topic in the news and everything today. It's really very interesting. There's so many good documentaries and videos out there. There's 3 in particular that I will post to your Blackboard site. One of them is called Resistance. It is awesome. I think it's like 45 minutes long, so it's not super long. It's on Netflix. If you have Netflix, you can watch it on there. I'm not sure if it's on Amazon Prime or anything Apple related, but definitely on Netflix. There's a couple others that are on YouTube. Again, one of them's a half hour, and it correlates with what we're talking about in this chapter so much. I think it's called Antibiotic Resistance, but I'll have it on your Blackboard page so you can watch it. Highly, highly recommend to kind of put all the pieces together and put a little real world spin to what we're talking about in here. So when you're looking at what drug resistance is when, let's say, for example, a particular antibiotic used to be very effective against a bacterial strain. All of a sudden, as time progresses, that same antibiotic is no longer effective against that bacterial strain. So somehow, that bacterial strain figured out a way to become resistant. How does it happen? It can happen due to some random genetic mutation. A lot of mutations are bad, but every once in a while there's a mutation which is actually good. And now the bacteria is, magically resistant. Or what may happen, which happens a whole lot, is the bacteria picked up its ability to become resistant from other bacteria. That happens all the time. The only thing that has to happen is the bacteria have to be close enough near each other where one can transfer its resistance properties to another. Does anybody remember where in a bacterial cell resistance properties are carried? The plasmids, yeah. So they're carried on the plasmid. And how are they transferred? Pili. Yep, so it'll be carried or transferred via that pili. So one bacterial cell is here, another one is here. That pilus, gets extended and it copies that plasmid, sends to the next bacteria. So now it's resistant. And they can do it very, very fast. One of the videos uses an analogy such as you have one resistant cell and then you have a bunch of bacteria that's not resistant, but in time all those other bacteria become resistant, almost like adding one red shirt in your load of whites. What's going to happen after that load? Everything turns pink because that one shirt leached out to all the other shirts in that load. That's what happens with these bacteria. One resistant one can send its information to everyone else, and now everybody is affected. Very, very scary at how easily bacteria can do this. So the more and more they're exposed to these drugs, the more and more of them are becoming resistant, and then they're sending it to each other. So again, how does it happen? Spontaneous mutations, okay, just a random mutation that's actually beneficial. That's more on the rare side of things. More commonly, they pick up those new genes from plasmids, from other bacteria. That on the plasmid, they call it R factors. It's called a resistance factor. That's the little piece that, it's a little piece that's on the, that's a little piece that's on the plasmid. Okay, that makes it resistant. It makes it resistant. So these R factors, some bacteria already have them. There's a bacteria that could have never encountered that drug, but yet it's still resistant to it. It just happens to have that information. So if it ever comes in contact with that drug is ineffective. A lot of bacteria just naturally have these R factors because if you think about it, bacteria growing in nature, when bacteria want to grow, they don't want any other bacteria around. They want to be the ones growing. They want to own that space, that environment, so they naturally have these factors to fight off other bacteria. What in the world does that have to do with antibiotics? If they're fighting off other bacteria, how does that relate to being able to beat an antibiotic? Yep, antibiotics are developed from bacteria and fungi. So if the bacteria has something to fight off other bacteria, that means that bacteria has something to fight off antibiotics made from that other bacteria. So they have these factors to fight off other bacteria. So therefore, they'd be resistant to the antibiotics that those other bacteria make. Awesome if your bacteria, really scary if us and we have an infection by those bacteria. I embedded a little video. It's about a 3-minute long video on here. It kind of sums up a lot of this stuff and a lot of stuff that we'll talk about. So I'll play that now. It's a cartoon, so it's kind of fun to watch, but it's really good.
What if I told you there were trillions of tiny bacteria all around you? It's true. Microorganisms called bacteria were some of the first life forms to appear on Earth. Though they consist of only a single cell, their total biomass is greater than that of all plants and animals combined. And they live virtually everywhere, on the ground, in the water, on your kitchen table, on your skin, even inside you. Don't reach for the panic button just yet. Although you have 10 times more bacterial cells inside you than your body has human cells, many of these bacteria are harmless or even beneficial, helping digestion and immunity. But there are a few bad apples that can cause harmful infections, from minor inconveniences to deadly epidemics. Fortunately, there are amazing medicines designed to fight bacterial infections. Synthesized from chemicals or occurring naturally in things like mold, these antibiotics kill or neutralize bacteria by interrupting cell wall synthesis or interfering with vital processes like protein synthesis, all while leaving human cells unharmed. The deployment of antibiotics over the course of the 20th century has rendered many previously dangerous diseases easily treatable. But today, more and more of our antibiotics are becoming less effective. Did something go wrong to make them stop working? The problem is not with the antibiotics, but the bacteria they were made to fight, and the reason lies in Darwin's theory of natural selection. Just like any other organisms, individual bacteria can undergo random mutations. Many of these mutations are harmful or useless, but every now and then, one comes along that gives its organism an edge in survival. And for a bacterium, a mutation making it resistant to a certain antibiotic gives quite the edge. As the non-resistant bacteria are killed off, which happens especially quickly in antibiotic-rich environments like hospitals, there is more room and resources for the resistant ones to thrive, passing along only the mutated genes that helped them do so. Reproduction isn't the only way to do this. Some can release their DNA upon death to be picked up by other bacteria, while others use a method called conjugation, connecting through pili to share their genes. Over time, the resistant genes proliferate, creating entire strains of resistant superbacteria. So how much time do we have before these superbugs take over? Well, in some bacteria, it's already happened. For instance, some strands of Staphylococcus aureus, which causes everything from skin infections to pneumonia and sepsis, have developed into MRSA, becoming resistant to beta-lactam antibiotics like penicillin, methicillin, and oxacillin. Thanks to a gene that replaces the protein beta-lactams normally target and bind to, MRSA can keep making its cell walls unimpeded. Other superbacteria, like Salmonella, even sometimes produce enzymes like beta-lactamase that break down antibiotic attackers before they can do any damage. And E. coli, a diverse group of bacteria that contains strains that cause diarrhea and kidney failure, can prevent the function of antibiotics like quinolones by actively booting any invaders that manage to enter the cell. But there is good news. Scientists are working to stay one step ahead of the bacteria, and although development of antibiotics has slowed in recent years, the World Health Organization has made it a priority to develop novel treatments. Other scientists are investigating alternate solutions, such as phage therapy or using vaccines to prevent infections. Most importantly, curbing the excessive and unnecessary use of antibiotics, such as for minor infections that can resolve on their own, as well as changing medical practice to prevent hospital infections, can have a major impact by keeping non-resistant bacteria alive as competition for resistant strains. In the war against superbacteria, de-escalation may sometimes work better than an evolutionary arms race.
Okay, so pretty good video, just kind of show you, some of the stuff we've talked about already, some stuff that we will talk about from here on out. My screen is blurry. Barbara, can you raise your hands up for a second? It's not working. Sorry guys, give me 1 second. There we go. Okay. All right, so looking at, in that video they showed how E. coli, like those, they were the fuzzy brown bacteria, all of a sudden just kind of, the antibiotic went inside the E. coli and just kind of like spit it back out. That's one way. It also showed another bacteria, that was able to produce an enzyme when penicillin came close. It just kind of like laser shot it away, so all of a sudden the bacteria or the antibiotic can't do anything. Those are examples of specific methods and how these bacteria are resistant. Because what does it mean that they're resistant? Like, what do the bacteria have that prevent the antibiotic from working against it? These are the 5 common things, okay, that bacteria have that would prevent a drug from working on it. So we're going to look at these items. This table is right out of your book, so you'll find that. So let's look at all 5. Okay, so the first one that I have up here is going to be an enzyme. We talked about penicillinase the day that we talked about enzymes. Remember, penicillinase is an enzyme that takes penicillin, so this is penicillin and I'm the bacteria. Here comes penicillin. I have an enzyme called penicillinase. I break it. Penicillin is useless against me. Okay. So, if you look at this, here's your penicillin. If the bacteria have penicillinase, it breaks this beta-lactam ring. Penicillin is now inactive. It doesn't work. Okay, so some bacteria have enzymes that they, employ just to destroy the drug. Penicillinase is in the class of beta-lactamases. Beta-lactamases are a group of enzymes that target the beta-lactam ring within these penicillins. Okay, so method number 1 is the bacteria has an enzyme. Okay, some type of an enzyme to destroy the drug. Next up, right here, in this scenario, big purple thing's the bacteria. It has these receptors, okay, on the outer surface, and these receptors, accept some substance normally. Okay, so it allows something to come inside the bacterial cell. Well, there's a scientist somewhere in a lab that designed an antibiotic that mimics this molecule. So the drug fits with the receptor, comes inside the bacterial cell. Bacteria doesn't realize it's a drug, and the drug kills the bacteria. Awesome. As these strains are becoming, they're adapting to allowing this drug inside, what they're doing is, well, if we change the shape of our receptor, the drug can't come inside anymore, okay? It cannot get inside the bacteria. So they just change the shape of the receptor. So method number 2, changing the shape of the receptor. This third one, this is called an efflux pump, okay, E-F-F-L-U-X, efflux pump. This is the one in the video that E. coli had, where it just kind of pumped the drug right back out. So here you have the normal receptors. Here comes the drug. It gets inside the bacteria, but the bacteria has a drug pump. It has this efflux pump, and it just pumps the drug right out. So you can keep administering the drug all day long, and the bacteria is just going to pump the drug right back out. So the drug is useless. The bacteria is resistant, the drug is useless. Next one, the fourth one is very similar to the first one, the second one. The second one at the top of your screen, the receptors became different. This one, the binding sites within the cell become different. Number 2 up here, that one was on the outside of the cell. This one, number 4, binding sites within the cell change. If the drug was designed to bind to the ribosome, that little site on the ribosome changed. Now the drug can't bind to the ribosome anymore. Last one. This is like looking at the reaction for folic acid synthesis as an example. So you have a drug that stops this intermediate B from making C, so you'll never get to the product. Great, so you administer that drug, it stops this reaction, no product. That would destroy the bacteria. However, the bacteria are like, all right, that drug is blocking this reaction, so instead, let's just make a different intermediate, take a different pathway, and we're still gonna get to the product. It's like an auxiliary route or a bypass route. Okay, so they're figuring out a way around, the function of that drug. Brilliant mechanisms by these little tiny microscopic bacteria for sure. Okay, so those are the 5 methods. Let's look at natural selection. The video talks a little bit about Darwin and the natural selection process. And again, what that video mentioned was, administering the drug, right away you kill off all the ones that are easy to kill. The ones that are not killed off are the ones that are tough to kill, but those ones that are tough to kill, if everyone else is gone, they're gonna grow like crazy. So then they take over. They are now the new population. Right now in your gut, at this exact moment, you have tons of good bacteria. You also have a handful of pathogenic bacteria in your gut. They aren't doing anything to hurt you right now. Why? Because you have all the good guys in your gut. Taking an antibiotic and it wipes out all those good guys, then those pathogenic ones in your gut right now can take over and cause an infection. Okay, they can become that new population. So we're looking at with natural selection, and we see that with this image in your text. So what's going on right here? Okay, so here we're looking at a population on a Petri plate. These white colored colonies, these are your, active, happy, easy to kill bacteria, good guys right here. They're not drug resistant in any way. So if you were to give an antibiotic, all these white colored colonies would die. These 2 red ones right here, these are resistant to a drug. But they're kept in check. They're really not doing anything because all these other ones are around. What happens when you administer an antibiotic? Well, it wipes out all these white colored colonies. These 2 are not affected because they're resistant, and then they will grow like crazy. So here, you add the antibiotic, and what starts to happen immediately, these are X's through the white ones. Okay, so all of these white colored colonies, the easy ones to kill, are gone. These 2 drug-resistant mutants are like, cool, we got all this space to grow. We get all these nutrients, all this space. In time, they completely take over and they are now the new population. Okay, so that's looking at this, theory of natural selection. The strong ones survive. The weak ones are killed off. Here's an example I just grabbed off the internet. It's just reiterating the same basic principle we've been talking about. This one uses Helicobacter pylori as an example. Does anyone know what Helicobacter pylori causes? Ulcer, yes. So pylori, think of pylorus, pyloric sphincter. Okay, so the distal end of the stomach. So Helicobacter pylori is responsible for ulcers. But here, you see the bacteria. Okay, so normal bacteria produces some type of an enzyme, whatever it is. Well, there's an antibiotic that targets that enzyme. So as long as the bacteria is producing that enzyme, the antibiotic kills the bacteria. Great. Scientists in the lab designed the antibiotic to target that enzyme to kill the bacteria. All right, but now what if you have a mutant strain of Helicobacter pylori? And now it doesn't produce the enzyme. Well, you give the antibiotic, and the bacteria is like, has no effect on me because I don't have that enzyme, so the drug is useless. And now this bacteria just keeps on multiplying, and now they all are resistant because none of them have that enzyme. Okay, so again, natural selection at play here. And of course, they can take that resistant information and send it along to other bacteria to make them resistant too. Some stats, I'll kind of throw some statistics your way that are a little bit alarming. as we go through the rest of the semester, or sorry, the rest of the lecture. So here you can see what percentage of prescriptions are actually given for viral infections, 75%, which is astonishing. I mean, that's crazy. So 75% of these antimicrobial prescriptions are for some type of respiratory infection. In most cases, it's just a viral infection, and these prescriptions are not necessary. Okay, so this is that shotgun approach. So take it, spread it out. Hopefully it takes care of the situation. I'm not really sure if it's viral, bacterial, whatever. This should work. But we know, using these antibiotics, yes, it can lead to resistance, but also it can lead to super infections. Again, the yeast infection example that I used before, that's an example of a super infection. Wiping out all the good bacteria, the bad ones like the yeast, bless you, can grow. This too is a huge problem. Okay, so here you're looking at a number of drugs produced in the United States, exported to other countries. Might sound like a fantastic idea, but looking at laws in some other countries, a lot of these antibiotics are just sold over the counter. And there's no rhyme or reason, they're just purchasing them and using them. Okay, it's not how it is in this country. So complete overuse in a number of other countries leads to an astonishing rate of microbial resistance. Hospital factor. Okay, so looking at the hospital factor, this is like the breeding ground for a lot of these resistant strains. Again, looking at patients in the hospital setting, most of them are on some form of an antibiotic. Okay, so most of them have, the easy to kill bacteria are gone. So that means the resistant ones are taking over and then passing it to healthcare workers who can pass it on to other patients, or the patient themselves passing it on to other patients. It's getting spread around, and you're looking at hospital-acquired MRSA popping up. I mean, there's community-acquired as well, but definitely the hospital factor is huge because everyone's immune system is compromised, and taking these drugs, you're wiping out these bacteria, and then the harmful ones are able to take over. Okay. And again, this is why, weakened immune systems, taking these drugs, workforce, you're looking at the staff maybe not following universal precautions and can spread, these organisms from patient to patient. But here we're looking at penicillin resistance in just about every single case of staph, in 30 years, nearly 100% of all Staph aureus strains, which is pretty crazy. Animal feeds, huge topic. It's such a controversial topic. There's so much at play with animal feeds, it's crazy. The politics involved with this is kind of insane. But this statistic is absolutely alarming. Okay, so nearly 80% of all antibiotics in the United States are given to livestock. Okay, so think about that. Okay. That's a whole lot of antibiotics just given in animal feeds. Why is that happening? At some other countries, it is no longer legal to administer antibiotics to animal feeds. This country, I know they're kind of working on that. It's really hard in this country because the reason it's given in the first place, okay, is prophylactic measure, okay, to prevent, maybe a farmer, I don't know anything about farming, okay, I'm not going to pretend that I do from Chicago, okay, I don't know anything. But as far as what I've researched and everything with farming, and I'm not talking your small, in-town farmer, I'm talking these large, super large companies. And, prophylactic means because you don't want one animal to get sick, because then it's very likely that they can spread it to other animals. And plus, these super large companies having just very unsanitary conditions with too many animals in too small of a space, where maybe you have chickens stacked on top of each other, so one gets sick and then is, defecating on others, and it's spreading throughout. Hog farms, whatever, once some of them get sick, it spreads very fast. Okay, so they're jam-packing them. And also, some of the antibiotics have also led to faster growth rate in larger animals, so you get more, meat for, from each animal. So that's also beneficial, looking at profit margins. And then you have, you know, like all the fast food chains that want to get their meat as cheap as possible, not caring where it comes from. They're going to go to places like this. Even though nowadays you're starting to hear a lot of places like antibiotic free. I mean, that's sort of coming more and more popular. But, you know, if you think about it from that farming standpoint, you want to have as many animals as you can so you can get more profit. If you have, you know, better sanitary conditions and have the animals spaced out, you're not going to have as many. There's going to be more work involved and so on. You're not going to, that profit margin is kind of diminish. Very interesting topic. One of the videos I posted, I think it's the resistance one, goes into this in pretty good detail, and it's really, really interesting. They talk about some farms in Denmark and what they've done, because it's completely illegal over there, but they've been super successful with eliminating the antibiotics from their feed. If you look at these antibiotic organisms that develop, so these animals are taking, you know, these antibiotics, killing all the good bacteria. There's some harmful ones growing within the animal, right? They're not too concerned about those harmful ones growing in the animals because the animals are going to be used to, you know, make some money. Great, but the problem lies with whoever's handling that meat from that animal, because now that meat is tainted with these resistant microbes, which if you mishandle them or if you don't cook them properly, those organisms you can get infected with. That's what's scary. If you go to the grocery store and you look at like turkey, ground turkey, for example, they said statistics are like 1 out of every 300 packages does not have drug-resistant bacteria in it. 1 out of every 300 does not. I'm not saying it does. It does not. Because there's one safe one out of that. Plus, they tested a handful of the ones that do have drug-resistant, and they found that a handful of them have what's known as pan-resistant salmonella. Pan-resistance means it's resistant to every single antibiotic that is out there. Okay, that is really, really scary, and that problem's getting worse and worse, and this is a huge factor with that. Okay, so not sure what's going to happen or what it's going to take, but again, there's a lot of politics in play with this, you know. Carly, do you have a question? Okay, some strategies. These points, they removed it from your textbook when they updated it. I kind of tossed it on the Prezi. I like to show it to students. I'm not sure why they eliminated it, but these are just strategies to kind of limit drug resistance. So here, when it comes to usage of the drug, it's up to the physician, you know, to prescribe the proper drug, right? I mean, it really is. You want to prescribe a narrow spectrum drug that targets just that bacteria. Is this done a lot? No. Okay, oftentimes broad spectrum. One of these organisms is probably causing it. This should take care of it, but it may also take care of a number of other bacteria and then lead to some resistance. Being a patient. Yes, you need to follow the rules of the physician, rules of how you should take that drug. What if you stop taking it too early? Well, think about it. As soon as you start taking it, what happens? You start to feel better. You're killing a number of the organisms that are present, right? So you're feeling better. Are there still some that haven't been destroyed yet? Yeah, and the reason they haven't been destroyed is because they're harder to kill. So you keep taking that medication and hopefully eventually you can destroy the ones that are harder to kill. But if you stop too early, well, you kill the easy ones. The ones that are harder to kill are still there, and now they're going to grow. So now you're going to develop a second reaction, which is going to be worse than the first one that you had. Okay, so definitely important to, you know, comply with the instructions with the medication. Okay, so here with some drugs, like I said before, administering 2 is oftentimes a good way to go. Okay, so here you have, at least one of them would be effective, and then a resistant strain may not be able to emerge. Research, just trying to figure out new drugs, right? Let's figure out some new drugs that don't have harmful side effects. Okay, let's work on that. And the last thing I have, long-term strategies. What's in here again? Education. Yeah, absolutely education. So people know how to take their antibiotics. A lot of this has to do with, overseas. Bless you. A lot of countries overseas. Here, what else? Okay, so especially valuable antimicrobials are restricted except when they're deemed absolutely necessary. So those like last ditch effort drugs, they're not given out like candy because you don't want the organisms to develop resistance against those last ditch efforts, right? You want to keep them, kind of safe. Let's see, stopping antimicrobials with animal feeds. That happening worldwide would be a good way to go. Let's see, government programs, vaccines. Okay, so all of this is just kind of long-term strategies that would help with this situation. Some new approaches that's happening. So probiotics are huge nowadays. You go into especially the dairy section, the yogurt section, you're gonna find all kinds of probiotic products. Do they all work very well? Absolutely not. Do they claim to maybe have the organisms that they say on the container? Maybe. Some do, maybe some don't. Maybe some did at the time of production, but by the time they reach the shelf, they are no longer viable. Or maybe if by chance they are active in here, can they make it past the acids and enzymes in your stomach to even make it to your intestines? Some can, some can't. I can tell you for sure that some of these products work better than others. Some of them don't claim, don't meet their claims. Particularly, I'll put my hand right there, but definitely do your own research, when it comes to that. They have probiotic pills as well you can take, but with any of that, just do your research. The goal, of course, is to replenish, bacteria within the intestines. Because taking a broad spectrum antibiotic, that's when you knock them all out. So you don't want to open yourself up to picking up a resistant strain or allowing an already present resistant strain to grow like crazy. You want to slow that down. The fecal transplants we talked about with C. diff patients a little earlier in the semester. So looking at an enema of fecal transplants just to replace that good bacteria. They also harvested it in a pill form. Doesn't sound very appealing, but that is another method that it can be taken, kind of dehydrated. The bacteria is all dehydrated and wrapped up in a pill, so you consume that. But this is something huge nowadays. And is it a good idea? If you are on a broad spectrum antibiotic, it is. They even have, for people that are prone to yeast infections, you take an antibiotic. There's some individuals that just always come down with a yeast infection. So is eating yogurt maybe the best means for that? Because, where does the yeast infection happen? Okay, within the vagina. So they do have lactobacillus swabs where you can administer the bacteria, in the vagina, in the exact area, which would be more beneficial than this, for example. So think about what's going on and think about what your desired outcome is and so on.
Does drinking alcohol destroy all the natural good bacteria in our stomachs and intestines?
No. It's not going to destroy all of them. Absolutely not.
But a lot of them, or no?
It can have an effect on some, but
I take probiotics every day, and I always thought that when you drink alcohol, alcohol destroys bacteria. So I thought not all of it, but most of it. And that's why.
What you're drinking is not
what's on the counter.
super strong.
I know.
Like the 95% what we have on the lab tabletop. It's and diluted with everything else. It's not going to wipe them out by any means. Does it have no effect? I'm not going to say that, but it's not anything to be worried about having, a couple of drinks here or there.
I wasn't worried. I just wondered what damage it actually did or didn't do.
Yeah, with the bacteria and alcohol, it's probably one of the last things I would probably be concerned with. Drinking alcohol while taking antibiotics, that's a whole new thing. You'll hear oftentimes, and it's really not true, that the alcohol interferes with how the antibiotics work. It really isn't the big issue. Does it not interfere? I'm not going to say that it doesn't at all. I mean, very, very slight, but not dramatic. The biggest concern with drinking alcohol when you're on an antibiotic is side effects. Okay, it's just going to amplify the potential side effects that go along with that antibiotic. Okay, prebiotics, basically consumption of nutrients that bacteria like. Okay, so then you're helping the bacteria grow within your guts. Okay, so that's what a prebiotic is versus a probiotic. But awesome, awesome idea when it comes to just keeping normal flora in check. Okay, so we got a concept check, so true or false? Bacteria can be resistant to antibiotics without ever encountering the drug. Yeah, absolutely. It's just in it with some, it's just some bacteria just have that. You should be able to list and describe the 5 mechanisms of resistance. What's an example of one?
Enzymes.
Having an enzyme like penicillinase. Maybe there's a drug pump that pumps it out. They change their receptor or their binding site. They can take a different pathway. True or false? Majority of antibiotics prescribed to humans are for viral infections.
True. 75%.
That's true. So 75% was the stat. And then nearly 80% of all antibiotics in the US are given to what?
Animals.
Animals. And that trend is just kind of, has increased over time. Once they start to see that, hey, I can have more and more livestock as long as I'm, administering the antibiotic, get more, profit from that. That is good if you were the farmer. Okay, just a couple of things to talk about, and then we'll just kind of wrap up the chapter. So we just need to talk about side effects and superinfections. So looking at side effects, we're going to look at just a couple examples of toxic side effects with some of these drugs. And we'll also talk about allergies, again, which we already kind of covered today. So here's a nice table in your book. Okay, so here you're seeing, major adverse toxic reactions to common drug groups. I circled a couple. Okay, I'd like you to know the couple that I circled. I know this is hard to read up here on the screen, but what I circled was the group of tetracyclines. Tetracyclines are known to discolor the enamel of teeth. And this is, if someone is pregnant, this could affect the teeth of the baby, as the child develops. Also, for kids, I think it's like under 8 or under 10 years of age, shouldn't have tetracyclines because it can affect their enamel. Okay, but if you're older, fine. Okay, tetracyclines, aren't going to have an effect on the tooth enamel. It could be pretty bad. I was actually at the dentist a couple months ago, and I had some recession, some gum recession going on in the back, and she's like, "Oh, we can do a very intensive cleaning. We'll numb you up with Novocaine, or we can just apply some tetracycline directly, into the gums." My first thought was tetracyclines, they're putting it right on my tooth and thinking this, but then no, older than 10, it's not going to have any effect on my teeth. And so, yeah, so it's known for that. It's one of the things. I actually have a picture. I'll show you real quick on the next slide. So here you can see this banding. You can see the dark lines. And this is a really good picture, okay? This is like an extremely mild case. If you were just to go to Google and type in tetracycline tooth enamel, you're gonna see all kinds of crazy images. This one's very mild from your book, but some of the banding is ridiculous. We're gonna see, complete brown and yellow discoloration. And oftentimes when it first happens, it's relatively yellow, but just exposure, to ultraviolet rays outside, it just darkens and darkens and darkens. It turns more of a brown color. Something else, and of course we can see, diarrhea. Okay, it's going to affect the colon. You see that with a number of them. The sulfa drugs, what we see here, you see they can affect the red blood cells. Okay, so hemolysis, they can cause some of the red blood cells to burst, and then also affects the kidneys, which you see with a lot of the antibiotics. Allergy is basically your immune system's working too good. It's working better than it should. It's, your immune system is meant to target and destroy these harmful invaders. Well, with an allergy, it sees something which may not be harmful, but it sees it as a harmful invader, and it mounts this gigantic attack against it. So it doesn't really know what it should be attacking and what it shouldn't. Okay, so like we talked about before, first exposure, that's when your body becomes sensitized and it starts building these antibodies. Like, if I ever see it again, I'm going to mount this gigantic attack against it. So second exposure is where you see that gigantic attack. And, pickup of these allergies, a lot of them, drug allergies are a little bit different, but a lot of them can happen. So maybe someone won't have allergies until they're like in their 20s or something. There's not a lot known as to the whys with this. Okay, so it could be environment, genetics, just, happens. And some people can outgrow them, so to speak, other times no. Not talking about drug allergies, but my cousin, when he was born, he was allergic to anything from a cow. Okay, so basically anything that had milk in it, you couldn't even eat a piece of chocolate and touch him. Completely break out in hives. Got better as he got older, like in his 20s, he was a little bit better. And, he'd go to McDonald's and be like, all right, don't use a spatula that touched any cheese, and he could eat a burger, okay, as long as there's no cheese on it. Who knows what's in the meat at McDonald's, right? But he was able to eat, burgers and be fine. Well, it was like last week, it was just before the holiday, they had, I think it was Potbelly, brought in, to work or something like that. And he had a sandwich which supposedly was okay, but it wasn't, and right away, just complete, throat swelled up, complete hives from head to toe. Thought, as he went on, he was better and better, but it wasn't gone yet, So he did get better, but then, it's just, it can, they're tricky. It's really hard to tell. So different types of reactions. Some people can have, like the hives, like skin rash, other ones, some type of respiratory, ailment, obviously inflammation, anaphylactic shock, of course, which you're looking at can be fatal. Looking at the most allergies, penicillins, obviously taking the cake with, the most amount of drug allergies. Out of all the drugs out there, the penicillins are the most common that people are allergic to. And it may just be because they were around for a really long time. It could be it. And it could be most prevalent. Don't know. It's just, again, allergies are kind of a mystery. All right, so the biota, or your normal flora. So your good bacteria that you have, they're everywhere. You want to keep them in check. So what I talked about before, you wipe out those good guys, bad ones can start to grow. You want to avoid that. So probiotics would help you avoid that. Taking the most narrow spectrum drug you can take would obviously help avoid that. It's these broad spectrum drugs that, tend to wipe everything out. Okay, so broad spectrum kills the agents, but also kills the good guys. Okay, that's not good, because you want those good guys around. And that can lead to a superinfection. Superinfection is that secondary infection you got because you were taking a broad spectrum antibiotic. Okay, and the example I put up here is that yeast infection, which we already covered today. Okay, basically the lactobacilli in the vagina keep, if you look at lactobacilli, so lactobacilli is found in yogurt and it undergoes a type of fermentation. Does lactobacilli produce acids or alcohol?
Yes.
Acids. Okay, lactobacilli produces lactic acid, which keeps the vagina more on the acidic side, which is awesome because yeast hate that. There may be a couple cells of yeast present, but they're not going to grow as long as the lactobacilli is there, keeping the vagina on the acidic side. As soon as you take something and you kill these guys, the pH of the vagina goes back to neutral. And once it hits that neutral, the yeast love it. They're going to grow like crazy. Okay, so good bacteria, they're there for a reason. Shouldn't try and eliminate them because then, more harmful ones can take over. And this just shows an example of a superinfection in the gut. So here you have a patient, okay, they have a throat infection. If you were to look at the status of their intestines, these white color colonies, these are the good guys. There's a couple pathogens present in the gut. So taking something broad spectrum, for the throat, as time goes on, it's working on the throat infection, but it's also circulating throughout the body, and it kills the good guys in the gut. So these harmful ones in the gut, once the good ones are gone, they can grow. And look, now they grew like crazy. Now this patient has a secondary infection in the intestines, but the throat infection is cleared up. So that new infection that emerged is called a super infection. And looking at something like C. diff, it's a spore former. So you wipe out the good bacteria due to taking broad spectrum antibiotics. Okay, and one of those spores happens to get into the gut, happens to get into your intestinal tract. Everyone else is diminished. They're able to grow like crazy and take over. Okay, so you see a lot of these C. diff cases with resulting from broad spectrum antibiotic use. Okay, so you should be able to describe the toxic effects or the side effects of tetracyclines. What do we see with tetracyclines? Tooth enamel, diarrhea, yes. Sulfa drugs, sulfonamides.
Red blood cells.
Yep, they're going to affect the red blood cells, and what else?
Crystals.
Kidney crystals, good. What group of drugs accounts for the highest number of drug allergies?
Penicillins.
Penicillins, awesome. Overgrowth of certain microbes due to antibiotic therapy is known as what? Superinfection. Superinfection. All right, so that is it for today. What I'll do is I'll post links to those videos on Blackboard. Again, I highly recommend that you take the time to watch them. If you watch any of them, I'll put this on Blackboard too, but definitely watch the one on YouTube, which is titled Antibiotic Resistance or Antimicrobial Resistance. I can't remember what it's called right now. One or the other, but that one is really, really good. So Michael, can you hang out for a minute? Jimmy, can you hang out for a minute too?
Yeah.